Hello everyone. Today we are going to start a new topic, that is plasma membrane. In this part, we will discuss about lipid component of plasma membrane. So let us start. Plasma membrane, it is outermost boundary of cell. Cell is the smallest unit of life. It is structural, functional and biological unit. Plasma membrane is selectively permeable. That means it does not allow the movement of all the molecules. Only selective solutes and solvents are allowed to move, and in this way it controls the molecular trafficking and thus maintains the internal composition of cell. There are various models proposed by different scientists to explain the structure of the plasma membrane. Among them, the most accepted is the fluid mosaic model proposed by Singh and Nicholson. It is most accepted. According to this, proteins like mosaic are embedded in the lipid bilayer. This is lipid bilayer. In this, proteins are embedded like mosaic, and plasma membrane also shows mobility, thus it is fluid in nature. Dynamic fluid like structure. Plasma membrane shows mobility. Lipid shows more mobility compared to the protein because proteins are larger molecules and have more molecular weight. Chemical constituents. Plasma membrane is basically composed of lipids and proteins. In addition to lipids and proteins, it also contains carbohydrate in small amount, that is approximately 5 to 10%. This carbohydrate is present in the form of glycolipids and glycoproteins. When the carbohydrate is bound to lipids, it is known as glycolipids, and when it is bound to proteins, it is known as glycoproteins. Carbohydrate is present in abundance in the plasma membrane of eukaryotic cells. Now let us see about the lipid component of plasma membrane. There are three types of the lipids present in plasma membrane. One is phospholipid. This is the major lipid component. Second, glycolipid. And third is sterols. The first is phospholipid. These phospholipids are amphipathic molecule. That means these have polar head and non-polar tail. This is polar head and this is non-polar tail. Polar head is hydrophilic. That means it is water loving or soluble in water, and non-polar tail is hydrophobic. It is not soluble in water or water hating. This shows the kink in the fatty acid chain. That means it is unsaturated or double bond is present. Now, how the phospholipids are arranged in the plasma membrane. Phospholipids are arranged in the form of the bilayers. These are present in the form of the bilayers. This is one leaflet and this is another leaflet. Here the non-polar tail are facing each other and this forms the hydrophobic interior of plasma membrane, and the polar head faces the aqueous phase, and this aqueous phase may be external or internal. It means that suppose this is the cell, this is cytosol, and this is the outside region. So this polar head, it is facing the outside of the aqueous phase and it is towards the inside. This is also aqueous phase, and these tails form the hydrophobic interior of the plasma membrane. The force that hold these two phospholipid bilayer is the hydrophobic interaction between the non-polar tail. Phospholipids. It is major lipid present in plasma membrane. It is of two types: glycerophospholipid and second is sphingophospholipid. In the glycerophospholipid, it consists of glycerol, then two fatty acid chain is present, and phosphate and alcohol is present. This forms the polar head, and this fatty acid chain forms the non-polar tail. In the sphingophospholipid, sphingosine is present one fatty acid, phosphate and alcohol is present. This is fatty acid, phosphate and alcohol. Here this forms the polar head, and this is non-polar tail. Glycerophospholipid. It is most abundant phospholipid in plasma membrane. Glycerol, two fatty acid chain and phosphate together known as phosphatidic acid. This phosphatidic acid plus alcohol forms glycerophospholipid. Depending upon the alcohol, the names are given. If the alcohol is serine, then it is known as phosphatidyl serine. If the alcoholic group attached is choline, then it is known as phosphatidylcholine. It is also known as lecithin. If it is ethanol amine, then it is known as phosphatidylethanol amine or cephalin. If the alcoholic group attached is inositol, then it is known as phosphatidylinositol. This phosphatidylserine is most abundant in plasma membrane. Lecithin and cephalin is abundant in plasma membrane of animals. This phosphatidylserine and phosphatidylinositol are negatively charged. This phosphatidylcholine and phosphatidylethanol amine has zero charge. So the charge varies. The second phospholipid that is present in plasma membrane is sphingophospholipid. It has sphingosine instead of glycerol, and one fatty acid chain is attached to the C1 carbon, hydroxyl group is attached, and this is known as the ceramide. That is the precursor of sphingophospholipid. Ceramide plus phosphate group plus alcohol is known as sphingophospholipid. When this alcohol is choline, then this sphingophospholipid is known as the sphingomyelin. Second type of the lipid that is present in plasma membrane is known as the glycolipid, in which sugar residue is covalently attached. Glycolipid is again of two types. One is known as the sphingoglycolipid. Second is the glyceroglycolipid. This sphingoglycolipid is also known as the glycosphingolipid, and glyceroglycolipid as glycoglycerolipid. So the structure is sphingosine it has one fatty acid chain and carbohydrate is covalently attached. In glycerophospholipid, glycerol to fatty acid chain and carbohydrate. In the sphingoglycolipid, to the ceramide, that is the precursor, carbohydrate is attached. Sphingoglycolipid is the major glycolipid present in plasma membrane. It is then classified into cerebroside containing single sugar residue and globoside which contains 2 or more sugar residue. This cerebroside is classified into glucocerebroside and galactocerebroside. Glucocerebroside if sugar residue that is attached is glucose, and galactocerebroside if galactose is attached. Then globoside. This globoside can contain sialic acid, that is with sialic acid, and it can be without sialic acid. If it is with sialic acid, then it is known as the ganglioside. This sialic acid is complex oligosaccharide. Known as N-acetyl muraminic acid. Third type of the lipid that is present in plasma membrane is sterol. It is amphipathic in nature. It is structural lipid. It is present in the plasma membrane of eukaryotes. It is rigid, planar molecule. It consists of 4 fused rings. Among them, the 3 rings are 6 carbon and the 1 is 5 carbon. This is known as the steroid nucleus. To the third carbon, first, second and third, hydroxyl group is attached. Derivatives of sterols that are present in plasma membrane. First is cholesterol. This is present in plasma membrane of animals. Second, ergosterol, plasma membrane of fungi. Stigma sterol, plasma membrane of plants. So this is all for today. In the next part, we will discuss about some of the important characteristics of plasma membrane. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.